We have already studied about the tools and processes of biotechnology. But what is the main purpose of developing these technologies? Biotechnology mainly focuses on large-scale production of many different industrial products. The applications of biotechnology includes therapeutics, diagnostics, genetically modified crops for agriculture, processed food, biomediations, etc. In this session, we will study the applications of biotechnology in agriculture. In the field of agriculture, biotechnology focuses on the increased production of food crops. Food crops production can be basically increased by three methods. First, by agrochemical based agriculture. Second, by organic agriculture. And third, by genetically engineered crop based agriculture. For agrochemical based agriculture, Green Revolution was initiated in India to increase the food production. It has succeeded in increasing the production by 3 times by using high yielding variety seeds, better management practices, and most importantly by the usage of agrochemicals. So, what do you think? Is this method cost effective for the farmers? The agrochemicals or high yielding seeds are very expensive. Hence, they are not cost effective for the farmers. But the usage of agrochemicals leads to harmful effects on the environment. Hence, the genetically modified organisms, that is GMOs, were developed. Well, why are the GMOs produced? The GMOs have many different adaptations. They are more tolerant to abiotic stress. They are resistant to many chemical pesticides. GMOs have long shelf lives, hence it helps to reduce post-harvest losses. They have increased mineral usages. They have enhanced nutritional value of food. GM has been used to create a tailor-made plants to supply alternative resources to industries, etc. In this session, we will study in detail on how biotechnology is used to develop pest-resistant plants. Have you ever heard about the term Bt toxins? Bt toxin is a kind of toxin produced by Bacillus thuringiensis, a bacterium. This toxin is a natural pesticide and hence used as a biopesticide. Now, what does they do to plants? They provide resistance to insects without using insecticides. There are many crops that has been grown by expressing the Bt toxin gene in them. For example, Bt cotton, Bt corn, Bt brinjals and Bt rice. Let's now see how Bt cotton is produced. As I told you, Bt toxins act as a pesticide. So, does it kill all types of pests? No. Bt toxins kill certain kinds of pests such as lepidopterans, means budworms, armyworms, etc. Coleopterans, means beetles, and dipterans, which includes flies and mosquitoes. The bacteria produce these toxins in inactive crystalline forms. When an insect consumes leaf containing these toxins, the toxins in their guts become active by solubilizing the crystals due to the presence of alkaline pH in their guts. Once the toxins get activated, it binds to the epithelial cells on the surface of the midgut. The toxin eventually creates pores that causes the swelling and lysis of the cells by which the insect dies. As said earlier, this toxin acts on specific insects only. Many different types of toxins are expressed in plants depending upon the insects that attack them. The genes that encode these toxins are commonly named as Cry genes. There are Cry1AC gene, Cry2AB gene that controls cotton bollworms, Cry1AB gene that controls corn borer, etc. This was about the Bt toxin resistance. Let's now see the RNA interference method to resist pest. We know that a number of parasitic nematodes attack plants and animals. One such kind of nematode is Meliodogenes incognita that infects the root of tobacco plants. The infected roots cause huge decrease in the crop yield. The process of RNA interference is used to prevent the infection. RNA interference is seen in eukaryotic organism as cellular defense. Here, a complementary strand of double-stranded RNA binds with the specific site of mRNA, silencing the mRNA. So, from where does this complementary double-stranded RNA come? The source can be an infection by virus having RNA as the genetic material or transposomes that replicate via RNA intermediate. The nematode-specific gene was introduced into the plant by a natural plant vector, that is Agrobacterium tumefaciens. This gene produces both sense and antisense RNA. Sense RNA is the RNA produced by the coding sequence and antisense RNA is the RNA produced by the non-coding or template strand of the DNA. These RNAs being complementary to each other forms a double-stranded RNA that initiates RNA interference and silences the mRNA. The result of which is the failure of survival for the nematodes on the plant host induced by this gene. Ultimately, the plant protected itself from the parasitic nematode. So, this was about control of pest using biotechnology approach. A drastic increase in the field of crops was seen due to the usage of these technologies. We have also seen that alternations of genes are very beneficial for humans. But is the alterations of gene in an organism beneficial for the organism itself? Try to get the answer. Let's now summarize what we have learned.
Manorama Horizon. Learning made simple.